And we are starting. Okay, theoretically, we are starting now. We're starting with the excavata. Starting with the excavata, we finished talking about the alveolates, which are part of this big SAR group, and the alveolates include apicomplexans. They also include ciliates and dinoflagellates, which do have a few parasitic members. Not hugely many, but some. And then excavata is another big group of eukaryotes that's not particularly closely related to animals, plants, or fungi. Most of them have flagella. Many have a specialized feeding groove. Many, but not all, have lost their mitochondria but retain evidence that they used to have them. Most of them retain modified mitochondria called hydrogenosomes that ferment pyruvate instead of putting it through the Krebs cycle. Free-living excavates. There's also lots of pathogens and symbionts. And the group we started with is the Euglenozoa, and that includes a lot of free-living little swimmy single-celled protists. Euglena is a common one that you'll see in pond water around here, but it also includes the trypanosomes. And the big killer in the trypanosomes would be African sleeping sickness. There's several others. There's a trypanosome that causes something called Chagas disease in South America, and species in the genus Leishmania cause pretty severe disease in South America and also in Central Asia and the Middle East. The Euglenozoa is related to Heterolabosia, which includes the infamous brain-eating amoebas that we'll talk about in a bit. And then the second subgroup of excavata, the Metamonada, includes the Fornicata, very funny, which includes Giardia, which is something that you most certainly can get around here and should probably be aware of. And two other groups, the Preaxostylans and the Parabasalians, that include some parasites and an awful lot of insect symbionts. And we'll probably get to those on Friday. So I've already talked a lot about Trypanosoma brucei, the causative agent of African sleeping sickness. It's spread between humans by the bite of the tsetse fly, Glossina morsitans, which was actually the subject of a poem by the great American humorous poet Ogden Nash. I'll pause this for a minute because somebody needs to get in, and I'll wait for our... Okay, there we are. Okay, we have our person has joined just in time for
Dr. Wagner.
The famous poem by Ogden Nash. Yes?
I was not sent the link again.
Yes, what's up?
I wasn't sent the link.
Again?
Yes. I had to get it forwarded from somebody else.
Okay. Okay, I'll check on that. I was sure you were in the address list. I'm not trying to leave you out, but okay, I'll make absolutely sure it was on the appropriate list. It's not in your spam folder?
No, I checked.
Okay. Okay, maybe the I don't know. I've heard the internet's infected with mailer demons. Maybe it was one of them.
Maybe so.
Yeah.
I just want to let you know.
Sorry, I don't know what. Okay, I'll recheck my address list and make sure it's got the correct information. All right, glad you made it in. Anyway, I was just recapping about trypanosomes, and we've already talked about African sleeping sickness, whose vector was the subject of a poem by Ogden Nash, who wrote, "Aunt Betsy was making out her will, and would have left us out in the chill. A Glossina morsitans bit Aunt Betsy." Tsetse. That's it. Sorry, it's the only. I read you the poems written by Ronald Ross when he solved the problem of the malaria life cycle, but that's the best I could find for tsetse flies. Anyway, I've said all I'm going to about Trypanosoma brucei and African sleeping sickness. In South America, they've got Trypanosoma cruzi, which is spread by blood-sucking insects. They are hemiptera, meaning they are true bugs related to stink bugs and things like that, in a subfamily called the Triatomini, affectionately known as kissing bugs. They're bloodsuckers, but they don't actually spread trypanosoma through the bite. They spread it through the feces. They'll pierce your skin and suck your blood and then poop, and you go, ow, and you scratch, and lo and behold, you've just inoculated your own wound with bug poop. And Trypanosoma cruzi gets into the bloodstream. This can last for decades. It's a chronic disease. No one is quite sure, but we do know that Charles Darwin spent much of his adult life with bouts of some debilitating illness that disturbed his digestion and left him weak and exhausted, pretty much for much of his life. And nobody knows exactly what it was, but it has been suggested that what it actually was that he got this during the voyage of the Beagle to South America. We know he was bitten by one of these kissing bugs, and it's suspected that he might have contracted Chagas disease. Anyway, the other big genus of Trypanosoma, Trypanosomatids, as far as human and veterinary parasitology is concerned, is Leishmania. And you get Leishmania from the bite of infected sandflies. In the gut of the fly, Leishmania exists as a promastigote, meaning it's an elongated cell that has a single flagellum sticking out front. Unlike sperm, where the flagellum pushes from behind, the flagella here in a promastigote like Leishmania pulls it from ahead. And these live in the gut of the sandfly, divide in the gut. They migrate to the proboscis, and the sandfly injects them into the skin of a human that it bites. In the old world, it's spread by sandflies in a genus called Phlebotomus, which is seen on the left. On the right, that's Lutzomaya. That's a different genus of sandfly, and this is the one that spreads it in the New World, in Central and South America. Once it's injected into a vertebrate host, Leishmania transforms from being promastigote into being amastigote, which means it doesn't have any flagella at all. Okay, sorry, just checking to see because I see somebody joined. It transforms into a stage that has no flagella. It's an amastigote. And these penetrate inside vacuoles in host cells. Macrophages in particular, the most important type of white blood cell, cells in the liver, spleen, and the bone marrow. They'll actually survive inside host cell vacuoles and reproduce there quite nicely. You're looking at a couple of macrophage cells You can see the large grainy nuclei and then all of those little dot things that look like little footballs with a darker spot inside surrounding those two nuclei. Those are Leishmania. And they're coated with lipophosphatoglycans. So I'm checking again to make sure nobody has joined or left us. And this actually stimulates white blood cells to ingest them, but it blocks the cells from digesting them. So macrophages and another class of white blood cell, neutrophils, will engulf Leishmania, but the same coat that stimulates them to engulf Leishmania blocks the cells from digesting them. And what would be a food vacuole becomes a parasitophorous vacuole. That phoros just means carrying. So a parasitophorous vacuole is just a vacuole that's carrying a parasite. And Leishmania will just live in there, reproduce in there. And it can survive undetected for a long time. It's possible for it to be asymptomatic. If it causes symptoms, it gets bad. Leishmania donovani and the related L. infantum cause visceral leishmaniasis, which means it attacks the organs. Visceral means it goes for the organs. The common name for this is kala azar. Symptoms include fever, anemia, potentially liver failure. This can be deadly, and an estimated up to 400,000 people are in all through the tropics. This man has survived Kala Azar, but he's suffering from a secondary relapsing infection that's causing these skin growths. So definitely not something you want to get. 2 other species in the Old World and then 6 different species in the New World cause a different form. They cause cutaneous leishmaniasis, meaning leishmaniasis in the skin. And that causes some pretty severe skin lesions. Given enough time, they'll eventually heal. I believe the disease is self-limiting, but it can leave permanent severe scars. It can also attack not just the skin, but the mucous membranes, in which case it is mucocutaneous leishmaniasis, and that's potentially fatal. Turns out that, I just found this out from a paper published last year, an estimated 20% of US troops that took part in Operation Iraqi Freedom came home with asymptomatic visceral leishmaniasis. Sandflies were, of course, very common in Iraq and surrounding countries. A lot of soldiers got bitten. During the active phase of Operation Iraqi Freedom, there were actually entomologists. Believe it or not, that's a thing. Coming out there trying to come up with mitigation strategies to keep people from getting bitten by so many sandflies and assessing how many sandflies might be carrying visceral leishmaniasis. And despite their best efforts, there weren't a huge number of active cases. I think it was only in the hundreds, but quite a lot of troops came home with asymptomatic visceral leishmaniasis, and that's concerning because it can be triggered much later. We know that immunosuppression, things like getting an organ transplant or chemotherapy, can trigger asymptomatic leishmaniasis to suddenly turn on you and become active. Alcohol abuse can do it, so can untreated diabetes, things like that. Anything that stresses your immune system can make that leishmaniasis suddenly turn on you and you get kala azar. Fortunately, it is treatable, but it's certainly something that veterans and the people that care for them are going to have to watch out for. And this wasn't the first time it happened. There were something like 1,000 active cases of leishmaniasis among U.S. troops that were stationed in the Middle East during World War II. There were something like 400 during the first Gulf War. I think the official count in the first Gulf War, the one where we took on Iraq and freed Kuwait back in 1991, there were something like 400 cases of cutaneous leishmaniasis and 12 cases of Kala-Azar. So it's certainly, a lot of the diseases we're talking about happen in far-flung parts of the world, and it might not seem like the sort of thing that concerns you directly. But even if you never go to a place where things like this are active, certainly there are people that do. This is the kind of thing that the military has to take notice of, because we often send our soldiers into places where there are diseases prevalent that are not prevalent in this country. Ecology and tropical medicine have actually been made by military doctors. Okay, right. So much for Leishmania. I've actually got a slide of it that we'll see in the next couple of labs. The other group in the Discoba, allied to the Trypanosomatids, is the Heterolobosians. And this includes some adorable critters called amoeboflagellates. And I did an undergraduate honors thesis with Dr. Stuart Bamforth at Tulane, wonderful man, on amoeboflagellates that live in soils. One in particular called Naegleria gruberi. And Naegleria gruberi creeps around in soils, particularly in the zone around plant roots. And much of its time is spent just as a plain little amoeba. But when conditions are bad, it can round up and form a resting cyst and just sit there. And then when there's lots of water present, it can transform into a little flagellate and go swimming off. I used to grow these on petri dishes, and if you flooded one with distilled water, in maybe an hour or so, you'd see little flagellates zipping about, and they'd swim around and then settle down and transform back into the amoeba stage again. Naegleria gruberi, the one that I worked with, is harmless. It grows at relatively low temperatures. But there's a species called Naegleria fowleri that grows in higher temperatures. It's very comfortable at human body temperature, and it can propagate in the wild in warmer bodies of water. Shallow ponds on summer's days and things like that. You can get lots of Naegleria fowleri. And it's normally free living, but it can be an opportunistic parasite if you get it up your nose. The next slide's an autopsy photo, so brace yourselves. If it enters the brain, it can go up your olfactory nerve, basically just start eating the nerve for your sense of smell and follow it back all the way to the brain. One of the early warning signs is loss of your sense of smell. Even if you go swimming in untreated water, it's not that big a risk. There aren't many cases of it each year. You really have to snort a lot of water up your nose to get this. And if you're ever concerned about it, the best thing to do is wear nose clips in the pool. Chlorination kills it, by the way, Cold water, it doesn't persist. We're talking about swimming in warm, untreated water. Although there have been a couple of cases of people getting this by using contaminated saline in their neti pots. If you know what that is, that looks like a teapot with a very long spout, and people use it for irrigating their sinuses. So anyway, once it crawls back up your olfactory nerve to your brain, it starts eating your brain, causing, at first it feels like the flu, then it feels like meningitis. And the problem is that it progresses very fast. By the time you're sick enough to go to the hospital, you're already in real trouble. And this is very rare. This is not something you really need to worry about, but it's almost always fatal. And we have an Arkansas connection here. There was a girl named Callie Hardig who got this at Willow Springs Water Park in South Little Rock, which is not open anymore. This is why. Well, this, and I believe 2 years previously, a young boy had caught this at Willow Springs Water Park and had died. She got it. She was treated at Arkansas Children's Hospital, and thanks to an experimental drug that was not approved in the US, but that they got emergency authorization for and flew in from Germany, Callie became the third person in 50 years to survive. I'm not sure what she's up to now. Her Facebook group, Prayers for Callie Leanne, is still there, but I checked it this morning. I haven't seen any entries in it since 2015. I'm told she did have a few deficits when she came out of the coma, but those eventually cleared up, and she's basically going on to lead a normal life. But still, this is rare, but it's not the kind of thing you would wish on anybody. So please do not go swimming in warm, untreated water without putting in nose plugs or something. Right. Okay, that was the Trypanosoma, or the Euglenida, and the Heterolobosia. The second group I'll talk about today, the second major group, is the Fornicata. There's lots of little free-living flagellates in the Fornicata that aren't very well studied. Ornithologists talk about LBJs, little brown jobbies, meaning small brown birds that are hard to tell apart. Botanists talk about DYCs, damn yellow composites, same thing. Lots of yellow composite flowers that are very hard to tell apart. Small colorless flagellates are kind of the protist equivalent. There's lots of little 10-micron flagellates that look like little bouncy balls under the microscope and turn out to be very diverse when you look at them in detail, but not that many people look at them in detail. Other than that, the main groups of the Fornicata are retortamonads and diplomonads. There's at least 2 retortamonads that live in human guts, commensal as far as we know. They don't seem to do any harm. The important ones that are parasites are in the diplomonads. Diplo means double. Monad means single thing, means unit. So a diplomonad is a double unit because diplomonads have paired nuclei and actually look like they're 2 cells that are stuck together. You'll see this in just a minute. The human retortamonads, this is one of them. It swims around in your gut as a feeding stage or trophozoite, makes cysts, and the cysts pass out in feces, and they're transmitted by the fecal-oral route. As far as I know, they're harmless. The name we use for the shape of the trophozoite is pyriform. You might see that word again. It means shaped like a pear. A retort is the name of an old type of glassware that was used for distillation, kind of like those heating bulbs that you might have used in chemistry classes if you did fractional distillation or anything like that. So anyway, these, as far as I know, are harmless human commensals. So is the other human retortomonad, Chylomastix, which infects an estimated 3.5% of the human population. It's also considered harmless, but it can occur in the same environments that human parasites like Giardia do. And if you have lots of it in a human community, it can be a general indicator of poor sanitation. People are drinking water or consuming food contaminated with fecal contamination. So something you might see down the road, those of you going into medicine, but not something so far that we worry about much. What we do worry about is the diplomonad Giardia lamblia. Giardia lamblia causes an intestinal infection called giardiasis, also known as hiker's diarrhea. It is not pleasant. I have never actually had this, but it's pretty miserable. You produce copious quantities of diarrhea. I usually see words used to describe it like foaming. This is not something you want to get. The cells are pretty small. They're only about 10 microns. And the one on the left, you can actually see it's got 2 nuclei. They look a little bit like a pair of eyes. And there's a midline formed by some flagella that I guess you could say looks like a nose. And then there's a little hook-shaped body in the middle called a median body that looks a little bit like a, if you're on drugs, looks a little bit like a mouth. This is what people look for if you're looking at fecal samples. You're looking for little 10-micron things that are smiling at you. It looks like a smiley face. Have a nice day. Have a foamy day. I'm Giardia. The picture on the right is a scanning electron micrograph, and you're looking at the underside of the cell, and you can see on the underside it's got what almost looks like a suction cup. That's what it uses to attach to the lining of your intestine. There's another view of it. You're looking at a ventral view right there. The total flagellate body is kind of pear-shaped, but up at the front on the underside, you can see that big sucking disc. If you look at it in cross-section in pictures B and C, that sucking disc is supported by a rigid arrangement of microtubules and microfilaments. The disc itself is pretty rigid, but the outer rim is flexible, and that's what Giardia uses to attach. You can see some Giardias clamped down on the gut lining in picture D down at the lower right. I'll show you a better picture of what this looks like in just a bit. Okay, you see the paired nuclei. The flagella emerge from basal bodies that are located between the nuclei. Some of them come right out, and then some of the flagella pass through the cytoplasm before they emerge. So those caudal flagella, the name appears down at the lower right. Those basal bodies are actually up at the front of the cell, but the flagella pass through the cytoplasm and then don't emerge until you get to the very back end of the cell. And it's those that create that midline, that create the nose, if you like. And then you can see the nuclei make the eyes. And last I looked, we're still not sure what the median bodies do. Another thing they're called is clawhammer bodies, because they look a bit like the head of a clawhammer, I guess. Right. And here's a whole bunch of them on the lining of your gut. They reproduce very rapidly. They can adhere to your intestinal lining in huge numbers. Unlike parasitic amoebas, they don't burrow any deeper into the tissues. They're not invasive, as we say. They'll just sit there on the lining, but they can cause a lot of damage in your upper small intestine. The duodenum is roughly the first foot of your small intestine, starting from your stomach. And then the section after that is called the jejunum, and it's there that Giardia hangs out. And you might know that the lining of your intestine has a lot of surface area because that lining is folded into lots of little projections called villi. The lining of your small intestine actually looks kind of velvety because instead of being a smooth lining, it's covered with thousands of tiny projections, almost like the little hairs on a piece of velvet. Giardia tends to make those flat, and that interferes with your intestine's ability to absorb food. And the symptoms are, or the condition that causes, this is a word you'll see again because there's other parasites that'll do this. They cause malabsorption syndrome, which is exactly what it sounds like. You're not absorbing digested food properly, which means that it can cause weakness, malnutrition if you're not getting certain nutrients that you need. And then, of course, everything that doesn't get absorbed by the small intestine has to pass through the large and then out. And that's the cause of the diarrhea. So that's another view of somebody's small intestinal lining that's absolutely carpeted with Giardia. They reproduce very fast. A severely infected person may be passing 14 billion Giardia in every bowel movement. If you're moderately infected, you may only be passing 300 million. Now, you can pass living Giardia, but they don't survive for very long outside the body. If you're looking at a stool sample from somebody who's been infected, you might see live ones, but the live ones don't normally go on to be transmitted. But Giardia also makes a cyst stage, and the cysts pass out in feces, and they get transmitted by our old friend, the fecal-oral route, usually transmitted by contaminated water, contaminated food, or by what we call fomites. A fomite is anything that transmits infection that isn't food. It could be anything from doorknobs to toilet seats to shared towels or something like that. Anything that transmits infection that you don't actually eat. Giardia duodenalis is known to infect over 40 known mammals. Now, we said earlier that parasites tend to be very host-specific, and this might seem to violate that. I've seen some suggestions that we're actually dealing with many species of Giardia that don't outwardly look different from each other, but are different on a molecular level. I'm not sure what the current state of knowledge is there, but we do know that a lot of mammals can serve as reservoirs. In particular, beavers are likely to be infected. And the reason this is called hiker's diarrhea is that hikers used to get it because even the most crystal clear flowing mountain stream can have Giardia cysts in it. Because, the rhetorical question, does a bear poop in the woods? The answer is yes. And the bear's poop can contain Giardia cysts that can contaminate water sources. Same for beavers or just about anything else. So you can drink water from what looks like a pure crystal mountain stream of the kind you would expect to see in a Coors beer commercial, and all the same get infected with this. And as I've said, it's pretty miserable. So if you're going hiking, even if it doesn't look like you need it, take those water purification kits. Use the tablets or the filters or whatever it is that you've got.
Those filter straws are great.
Okay.
Sorry.
My wife says filter straws are great.
Sorry to interrupt.
Mm-hmm.
He's asleep.
Yes.
Yes. He's exhausted. I don't think he feels very good.
Okay.
I've wrapped him up. He's asleep. I'm going to be out here for a few minutes working on some stuff.
Okay.
But I just wanted to let you know.
All right.
And,
Has he done as much schoolwork as he can right now?
he's done his class, and then he has to just go back to sleep.
Okay. All right.
Sorry to bother you.
Okay. All right. Sorry for that momentary interruption. Yeah, we're doing the virtual school thing, and yeah, it's kind of messing with my kid's sleep cycle. Anyway, more than you needed to know. There are some other diplomonads that infect other species. This one is Spironucleus. It is not known to infect humans, but it's the kind of thing you might encounter in veterinary practice. Spironucleus meleagritis is a parasite of turkeys. Infected chicks, if they survive, they don't grow to their full size. They grow up stunted. There's a couple of Spironucleus species that parasitize salmon. Spironucleus vortens, shown here, is a parasite of aquarium fish. So you might encounter other species of fornicatans, other species of diplomonads, those of you that go on to veterinary practice. And that would actually appear to be that. That is it for my slides. We'll pick up the last of the excavata on Friday. And I do believe we've got an exam coming up. I will try to post you guys some questions and things like that. And I've clearly got to do some thinking about the format. And I'll see roughly half of you tomorrow, and we'll go hunting for Neospora caninum, hopefully with slightly better results than the first group got. Nothing on the first group. They did fine. It's just the sample we had didn't have a lot of them, but maybe I'll try another one that might. And I will now stop the recording.